Hi, everybody. So my name is Wiley. Today I'm going to talk about looking at forest edge characteristics using NEON's Airborne observation platform. So my research stems from this idea of forest fragmentation, that as developed areas and deforestation are increasing over the whole landscape of the world, forests are becoming increasingly fragmented. Research has found that more than 70% of global forests are within a kilometer of an edge, around 20% are within 100 meters of an edge, and a little bit more locally, almost a quarter of northeastern US forests are within 30 meters of a forest edge. And so what I say when I mean forest edge is where the forest physically stops becoming forests. So whether we have a forest that turns into an agricultural field, or maybe a road or a housing development, simply where the forest simply stops becoming a forest. And other research has shown that forest edges have different microclimate conditions than forest interiors. So forest edges are slightly more susceptible to wind and drought and fire than forest interiors, for example, but they also have greater light availability and increased access to crucial nutrients such as nitrogen. And research has found that in New England forests in particular, trees towards the forest edge are actually larger than trees towards the forest interior. So they are able to store more carbon towards the forest edge than towards the interior, which has significant implications on regional and global carbon budgets. So my research kind of ties this together into look at a couple of different aspects of forest edges, looking at canopy height, looking at leaf level nitrogen content, and how these relate to forest edges. So there's kind of a dual hypothesis going on here, in that we know that forest edges have greater biomass than trees towards the interior, but we don't know exactly why. So one potential for this is because we have greater leaf area at the forest edge, which means we have more leaves that are able to sequester more carbon, and that is why forest edges have greater biomass than interiors. And the other reason is that the leaves at the forest edge are slightly different than leaves towards the interior, in that they're more photosynthetically efficient. And we can look at this by looking at leaf level nitrogen, for example, which oftentimes corresponds with higher chlorophyll content, which will correspond with higher photosynthetic efficiency. So in that case, we'd probably see a roughly the same vegetation density towards the forest edge, but just have structurally different leaves. I also wanted to look at whether remotely sensed values compared to in situ measurements. And what I mean by this is that we collected two different types of data over the course of last summer. One was in situ, which is where someone actually physically goes out into the field and collects data. So on the top here, we have canopy light transmittance, which is a camera that's placed in the forest and pointed upwards. And based on the amount of light coming through the canopy, you can tell how dense the vegetation is. And on the bottom, we have foliar sampling, which is someone goes out into the woods and collects leaves from trees and then takes them back to a lab and processes them for carbon and nitrogen content. And so the other type of data collection that we did was we actually used data from NEON, or the National Ecological Observatory Network, which annually flies a plane over the Harvard Forest in Central Mass, annually flies a plane over and gathers imagery. And this imagery contains a lot of information because for each pixel over the forest canopy, there's a certain light intensity for every wavelength in the electromagnetic spectrum within that image. So we can look at the intensity of light coming, and we can look at relationships between different wavelengths and determine canopy properties based on these relationships. So looking at nitrogen, for example, we can use the Normalized Difference Nitrogen Index, or NDNI, and look at a measure of canopy nitrogen based on the relationships between these wavelengths. And there's a kind of a big difference between these two different data types, in that one, you actually have to physically go out into the woods to collect data, and the other you can do from a distance. In situ data is oftentimes more difficult. It's more labor intensive, it's more time intensive, and oftentimes more expensive than remote data, which can get a lot of data very quickly. So it might be better for scaling up these measurements. So these are three plots at the Harvard Forest that we did research on. So on the left here, HFO3, we can see this is a forest edge that borders on an old agricultural field that's starting to grow back. HFO4 here in the middle, we have forest edges surrounding a current agricultural field. And then HFO6 on the right, we have a road plot right here where the forest edge is bordering on this road. And so for the sake of this presentation, we can just look at one plot. We can look at HFO4 as a case study for some of the research that we did. So HFO4, right here, we have a handful of different forest edges bordering this agricultural field. A house right here, this is the middle of the woods. And then with these lines right here that are plotted are GPS points of two different metrics of forest edge. So the yellow line represents the tree line or the stem line, which is where the physical tree trunks end. That's where the tree trunks stop right before the agricultural field. And then the red line represents the drip line, which is the canopy hanging over above the field, but doesn't have any actual tree trunks below it. So the first thing that we looked at was a canopy height model. And so this is done while the airplane flies over. In addition to having the imagery, it also has LiDAR, which is a pulse laser that shoots downward from the plane into the forest canopy. And based on the different returns of the laser, we can look at how high the tree is. And this works just by, when the laser comes down, slight portions of the light reflect back up to the airplane, but some of it also penetrates down into the canopy. And so then we can look at how long it takes the light that's returned from the top of the canopy versus the light returning from the forest floor. We can look at how long that time takes for that pulse laser to return to the airplane, which will tell us how high the canopy height is. And so we can see, just kind of looking anecdotally at this, that we tend to have higher trees over here on the west side of the plot, or on the west side of the field rather, versus on the east side of the field. But we wanted to look at this a little bit more continuously and kind of look at tree height as a function of the distance from the edge. So to do this, we ended up pulling data from each of these sides here, this west-facing side, this east-facing side, and this east-facing side, and then created buffer zones where every 2 meters, we created a new buffer to look at the canopy height within that certain distance. So 0 to 2 meters from the drip line, 2 to 4 meters, 4 to 6, so on and so forth. And that helped us produce a plot like this. We can look at the distance from the drip line and then the canopy height. The green line here represents the average distance between the drip line right here and then the forest edge right here. And we can see that we have lower canopy heights right towards the end of the drip line as the trees are hanging over the field. But then as we move into the canopy, we have a slight bump right here at about 20 meters, representing higher trees right at the forest edge. And then that does kind of level off as we move into the plot, but doesn't quite get as high as it does at the very beginning. Additionally, looking at canopy nitrogen, we gathered data within one transect of this plot. And this was done for a couple of reasons. One was because we could just look at exactly how far we were from the edge of the forest. It's very easy to just kind of take a tape measure and you knew exactly how far you were, whereas that might be difficult on other areas, especially if you're taking measurements from different edges, different distances. So we just picked this transect. And this was done not only here, but also at a handful of other plots around the Harvard forest, just so we had a more robust data set and we could kind of operate with different measurements. Rather than taking all of the measurements from one plot, we could take some measurements from a handful of different plots just to make our results a little bit more robust. And so in looking at canopy nitrogen content, we end up seeing an interesting pattern, which is that right here towards the forest edge, we actually have lower canopy nitrogen, lower leaf level nitrogen than we do towards higher in the plot, or towards further into the forest. And this was interestingly echoed in the remote sensing data. So NDNI, the normalized difference nitrogen index, we can see that we have low values right here, right at the forest edge. The pink line here represents the drip line, and then the red line right here represents the stem line. So we can see that in both cases, we have lower canopy nitrogen towards the forest edge. And so this is an indication that the leaves are not more photosynthetically efficient than leaves towards the forest interior, for example. So in looking at the vegetation density, we would expect to see a higher density towards the forest edge. And in fact, that's exactly what we did see. We can see here that canopy light absorption is a measure of how much of the light is absorbed by the canopy as a function of distance from edge. And we can see here that the vegetation towards the edge of the forest is able to absorb a lot more light than vegetation towards the forest interior, which just indicates that the vegetation is more dense, it's blocking out more light at the edge relative to the interior. So some conclusions here. We have loose agreement between the in situ and remote data. Again, we saw lower nitrogen values at the forest edge, both in the foliar sampling data as well as in the remote data. We also saw denser vegetation towards the forest edge and increased biomass, which goes along with one of our hypotheses from earlier. But if we're going to expand this to carbon accounting in all of New England, for example, or even globally, we need to be able to have a more robust data set, and it needs to be a little bit more generalizable. We're just looking at one plot here, but if we're really going to apply this to our carbon accounting that we do on a larger scale, it's possible. This shows that we have the ability to do that, but we're going to need some more estimates from different places around not only New England, but also around the country and the world if we're going to want to scale this up. Thank you.